Okay, so your reading entailed a concept called a spectrophotometry, and how spectrophotometry is used in conjunction with serial dilutions, so that scientists use these two, the combination of these two things, for a very specific reason. And so your reading should have elucidated those reasons or reasons. And today in class, we are actually going to talk about the specific research problem that we have. So this whole lab, it's online. Even if it was in person, I make these pretend research situations. And so the goal here is to help get you used to thinking like a scientist, not just doing what you're told to do in the lab, but actually starting to ask the questions, why am I doing it this way? How does this technique actually help me solve the problem? How do I do this technique? What are the reagents? Why do I have to use this reagent versus the infinity other options I have out there? So that's probably the biggest difference between lab in Bio 103 versus lab in 100 if you took that, 101 or 102. So it is my impression that the labs in plant and animal are, here's this thing, follow the directions, you're done for the day, don't ever think about it ever again, it doesn't matter. So that is definitely not the approach in this class. The approach in this class is, you are baby molecular biologists, and I'm starting you from the bottom, and we're going to fine tune those critical thinking skills. We're going to practice asking questions. Like when you're a little kid, if you have little kids or you have little siblings, whatever it is, what's their favorite question? But why? Go back to being a little kid. Always ask, but why? Okay. Oh, how, why does it do that? How does it do that? And these are the questions that a good scientist, if you want a career in science and you want to be in the medical field, the research field, the biology field, you need to not just accept what's given to you. You need to challenge it always. So we are doing that with a very simple exercise, but what this will show you is that even with the simplest tasks, you can ask the questions why and how. Okay, so let's share the screen. Okay, come on. Great, let's make this bigger. All right, so today, this is Wednesday, week 2 lab. And we are going to be discussing how to use serial dilutions and spectrophotometry in order to address a certain research topic, or a research problem or a research phenomenon. So one of the cool things about being online, and you're thinking like nothing's cool about it. Yes, there is, I promise you. One of the cool things about being online is that you can actually spend more of your time and energy, your conscious energy, on the other steps of the scientific method that most times get overlooked in person. Okay, so the scientific method should definitely be reviewed for you. So we're going to go through what the scientific method is, at least the first couple steps today. So it all starts with observing something awesome or confusing or something you want to find the answer to. So you need to observe. I also call this, Paloma, don't be starting your barking. Okay, so we observe a phenomenon, or we observe something really cool that excites us and interests us, or we identify a problem. Wow, that's a problem. What's a problem? Cancer is a problem. We need to solve this problem. What else is a problem? Any disease, any disorder is a problem, and we want to solve that problem. You can identify a problem. You can identify a question. Okay. How does skin form? I don't know. Okay, let's figure it out. Okay. That is the first step of every single thing we know about science. It all started with either observing this cool thing or identifying a problem or a question. So even this really simple activity that we're doing in this class, your job is to identify what the question is or what the problem is. Okay. And so the next thing that you want to do, so observe a phenom or something cool. Okay. So you've established what is interesting to you. And then what a scientist will do is they will come up with what they think the answer is, or why this thing is cool, or how this problem can be addressed, right? And so these things are called predictions. Another word to use for prediction is hypothesis. So you make predictions / hypotheses. And these are always based off of what somebody before you has shown the scientific community. So the way that we like to talk about predictions and hypotheses are that they are based on previous studies or rationale, previous findings, we'll call them. Okay. And that could be any incoming knowledge. So, if you are watering a plant with acid, because the question is, what would happen if I watered the plant with acid? Your prediction, you would say the plant would probably fry and die. And you didn't just pull that out of your mind, out of thin air. That prediction was based on what you know about acid and how acid can destroy things. So everything is based on what you've learned from other people. Those are previous findings. Okay. So good hypotheses, strong hypotheses will be based on strong and abundant previous findings. Okay. So not only do you come up when you're doing your predictions and hypotheses, you're not only going to come up with what is the likely outcome, but you also a good scientist will also establish, hey, in the unlikely event that something different happens, what are those different things? What are those different data? So what are the unlikely outcomes? Okay. Because you may not know it now, but if you do get into a career of science, most times this is what you get. Unlikely outcome. The outcome that you get isn't exactly match your hypothesis or your predictions. And so you don't want to be taken totally off guard if you kind of already suspect, okay, well, some other options that could happen from this experiment we're doing is instead of seeing an increase, we see a decrease. Okay, what is a decrease? What would that decrease mean? Or instead of seeing an increase, which we think will happen, it should happen, we see no change. No change. Okay, well, what does that mean? So if you're able to come up with multiple outcomes, you'll never be caught off guard, and you'll be able to describe or explain what those data mean. Okay, so next, now it's time to plan the experiment. You never just start doing the experiment. You need to have it planned out before you begin. So you need to have it planned out, make sure you have all your reagents, make sure all your equipment works. You want to have it all planned out on a piece of paper in your lab notebook. Okay, and every single great experiment, not even great, every single even average experiment better have controls, and they better be good controls. If you don't have controls in your experiment, that experiment needs to be flushed down the toilet. Okay, that is no good. No one wants that. So you need to plan your experiment, and the first thing that you need to plan or identify are your controls. Controls, you have known information about them. okay? You either actually know it or theoretically know information about it. So in parentheses, I'm just gonna write actual info or theoretical info. Okay, and those terms are important for what we are doing today. So I'll give you a clue that our controls, we have theoretical knowledge of controls. We don't have actual knowledge, we have theoretical knowledge. Okay, after you decide or determine your controls, you should also determine what the outcomes, predict the outcomes for these controls. Controls should be something that you put through the same experiment, you use the same equipment on them, and you should have an idea of what that piece of equipment, if it's a number, you should have kind of an idea of the number that should pop up. If it's a trend or a change, you should kind of see like, ooh, well, a negative control should have 0 molecules. A positive control should have a bunch of molecules. Or, oh, I'm going to be assaying for a trend. So if my controls work, I should see an upward trend. If my controls work, I should see a downward trend. So you should know the outcomes of your controls before you get started. Because the first thing that you should do is when you actually do the experiment and you're looking at the data, the first set of data that you look at are going to be your controls. And if those controls don't match your predicted outcomes, trash the experiment. Okay, that's the way science works. You don't just force your eyes to believe like, I think those controls, I think the data is, no, is it or isn't it? Don't try to convince yourself that your controls work. You need to have solid controls. Okay, so plan your experiment. First things, get your controls, predict the outcomes of the controls, know what you should see when you test the controls. And then you have your testable samples or experimental samples. Okay. These are the samples that you don't know anything about, or whatever data you're collecting, you don't know the data. Okay. So this is your some kind of unknown value or trait or outcome. You don't know it. You might guess, but you really don't know it. This is really what you're going to be making your hypotheses about, is your testable samples, the things that, those are your question marks. Okay. All right, so all these things, steps 1 through 3, these are all things that should be done in any lab, in any class, in my opinion, should be done. Okay, so after you plan the experiment and you gather all your equipment, you do the experiment. Okay, so this is the part where I say, I'm on your team and I did the experiment. So you're not physically doing it, but I went to the lab and I physically did it. Okay, so this is real science. I'm not going to give you some simulated cartoon on the computer to like move your mouse around. You're going to look at real science and you're going to analyze real data and you're going to make real figures and you're going to write about this real data. Okay, so after you do the data, what do? You collect data, or after you do the experiment so that you can collect the data. And you will do that. Not today in class, but today after class, or sometime after class, after lab today. You collect the data, you analyze the data, and then you explain what that data means. So the difference between analyzing and presenting data versus presenting conclusions. Here's an example. The data shows a positive trend. Okay, there's no information about what positive trend means. All you're saying is the data shows a positive trend between number of hamburgers consumed and weight on a scale. The more hamburgers you eat, the more the scale will read. There's your trend. Okay, but then the conclusion would be that you would make a conclusion about how eating hamburgers somehow activates the weight gaining, I don't know, cellular process, right? So you would explain what that would mean. So hamburgers have a high fat content, and so the fat is not metabolized, so the weight is maintained on the organism, which causes the scale to increase. You've just taken a trend and you've explained what that trend meant, okay? So you analyze the data, and then you make a conclusion. So the conclusion explains it. The analyzing part is almost like stating the result. Okay? Analyze is stating the result, explains, conclusion is explaining the result. Okay. Great. And then last but not least, now you need to do a good job of communicating that to the scientific field. Communicate your findings. So step 5, maybe you do that in your group, you do that in your lab notebook, you talk to your advisor, you maybe the hypothesis wasn't entirely right, maybe you want to go back and do the experiment again. Okay, fine. So in parentheses, you might say redo, revise, whatever, but you get back to the same place. When it's time for you to actually communicate that science, you're going to do that, and you can do it in one of two ways. You can do it in written form, and the written form is not going to only have words, but it's also going to have figures. Or you can do it in verbal form, audible, okay? I guess oral. So that's going to be a presentation, and the presentation is going to include your voice and also figures. Okay. Or interactive science, play. Okay, so if you can see from this list, I know like the typical scientific method has like a set number of steps, but I just kind of combine them, mix and match how it makes sense to me, but all the steps are there. You are going to be the scientist. The only thing that you are physically not going to do is step 4. That's it. So you're still acting like a scientist. And so what we're going to do today in class, so let's see. In class today, we are going to talk about these first 3 things. Okay. And that's what you're going to do in your first breakout room. So for the serial dilutions, spectrophotometry, blah, blah, blah activity, the first thing, well, I guess it doesn't have to be first, but it makes sense to identify the problem first, is with your group, talk about what you think the problem, the question, the phenomenon is. And so you need to come up with a statement that tells us, what is it that we're trying to, why are we doing this? Why did I tell you that we're going to do the serial dilution spectrophotometry? And if you said that that's just because it's in the syllabus, that's the type of thinking we need to change. Okay, so why did I decide we're going to do serial dilution and spectrophotometry? What is the question that I'm saying? We're going to use this approach to solve a problem. What is that problem? And then you want to make predictions. So what do you think, based on the problem, what do you think the findings will be? And so that needs to be based on something else. So I'm going to ask you guys to open up your chat. This is for everyone. So everyone should be engaged. And I'm going to ask people to contribute. What is spectrophotometry based on? You can just put phrases, you can put words. What do you think we're doing with spectrophotometry? Anything that comes to mind? Okay. Okay, cool. All right, so I'm going to just jot down essentially what you're saying so that we have it on this piece of paper that is being recorded. So spectrophotometry has to deal with, and I'm just going based on what you're saying, is the absorbance, which can also be called optical density of specific light waves. Okay, that's what a spectrophotometer does. It shoots little wavelengths of light determined by the scientists at a certain sample. Great, that's what the spec does. Okay, but as people are mentioning, scientists use spectrophotometry because it helps them in some way, shape, or form determine the concentration of samples where we don't know the concentration. Okay? Helps determine concentration. Concentration is just brackets. Okay. I see people are putting something along the lines of, usually when people are using the spectrophotometer to do this certain goal, they first need to do serial dilutions. So there's some serial dilutions going on. Okay, let's see what else we got. Okay, okay, now I'm starting to see why. Why do we use spectrophotometry? And whether or not you knew that you were answering this question by contributing to the chat, you actually did, because some people wrote the relation of light to concentration. So we reform that just a little bit. So essentially this is based off of some sort of relationship. Okay, and that relationship is between absorbance and concentration. Okay. So what is the relationship between absorbance and concentration? Okay, awesome. So you're all saying something, you're saying it in different ways, but it all means the same thing. Oh, yeah, Cole actually just summarized everyone's comments perfectly. So there is a direct relationship between the concentration of a solution and the amount of light it can absorb. And so what a direct relationship means is, as you increase the concentration of the solution, you also increase the absorbance. The direct, so it's direct. Direct means if you doubled the concentration, you should double the absorbance. If you cut the concentration by 5, you should cut the absorbance by 5. That's what direct means. So it's a direct relationship. You double one thing, that means the other thing should be doubled. That's in theory. In theory, that's what you should have. Yes, Ivan, they're proportional to one another. Great. So we all now are putting these pieces of information down so we can start seeing how and why a scientist would use spectrophotometry and serial dilutions to help them determine concentration. We do it because the relationship between absorbance and concentration exists. We know it. It's been established from many scientists before, and a scientist with the last name of Beer. Okay, so Beer's law really does establish this relationship between absorbance and concentration. All right. Okay, so this is your previous findings. It's called Beer's law. We're not going to get into the equation. That's for a different class. Okay, great. So we're already kind of talking about some of these things that you will need to further expand on in your group. Okay, so if that is the case, so the previous findings we kind of already took care of. Uh-oh, someone's at my front door. I'm so sorry. Give me 1 second. Okay, so in your groups, identify the problem or the question. Next, predict what you should see, okay, or what you think you should see, and that's going to be based off of what we said, the relationship between absorbance and concentration. So what should you see for the samples that you're going to be analyzing with regard to concentration and absorbance? Okay. those would be the likely outcomes or the typical predictions, and then maybe just one or two unlikely outcomes that you may see when you're collecting data. Okay? And then the third thing is you are going to sit down and plan this experiment. So the protocol was already written for you. It's in the boot camp manual. And essentially, I planned the experiment. Like, you know what to do, but some of the things that you would have to actually figure out. So the first thing that you definitely need to talk about is, what do you think your control or controls are? Okay, so again, I'm going to remind you, controls are the things that you know information about. Now, the information can be real life. You have it, it's like the concentrations on the bottle, so you know it, or it can be theoretical. So what are the controls and what are the predicted outcomes? Okay, so in this planning the experiment, we know that serial dilutions are involved. And that's kind of step one, right? So I'm giving you some guidance. Step one is performing the serial dilution. How do you do that? Draw a little picture, just like you did in the homework, that shows and explains how you do the serial dilution from the manual. So how? So you're going to include things like the dilution factor, how much of the previous solution to move, how much of the diluent do you need, how many tubes do you need to set up, what is the dilutions of all of those tubes, what blah, 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 okay? How, that's the how. That is the detail that I'm expecting. Once you do the serial dilution, what's next? What's next? Okay, for each of these what's nexts, I also want to know why. What's that going to provide you? So just a quick question to the class. After you do the serial dilutions, according to the protocol and the notes that you took, please enter into the chat, what do you do with those what do you do with those dilutions? Okay, good. So the next people told me was put them in the spec. What valuable information are you going to get when you put those samples in the spec? What info do you get? Yes. Okay, so hopefully that shows you what I'm looking for is you're going to do a thing so that you can obtain a valuable piece of information. And then with that valuable piece of information, so once you get the ODs, then you do something next. Don't put it in the chat, this is for your group. What is that next? And what is that going to provide you with? What do you use that for? Okay, this is the thought process that you have to start practicing and getting in the habit of using every single time you're in lab. Okay, this will also be beneficial for class. Once we start getting into these complex mechanisms, your questions will be like, well, why does it happen like that? Why not another way? What are those other ways? Okay, so you can definitely predict the outcomes of what you would see, right? If you're looking for a trend, you should be able to predict the trend of these serial dilutions. And then you have testable samples. So identify what those testable samples are. It's in the protocol. Okay. And that's pretty much it. Okay. So those are the things that you need to talk about in your breakout room. I gave yesterday's class 35 minutes. I think that was too long. So let's go with 30 minutes. I think that that should be enough time, and I'll give you a 10-minute warning. And so if you feel done in like 10 minutes, that would tell me you don't have enough detail. You don't have enough answers to whys and hows. So go back and keep working and just keep asking why and how and get the answers to those. If you're done in about 25 minutes, then probably you're done, and you can, take a break until the 30 minutes are over, or you can, I don't know, do whatever you want. Okay, talk to your new group, great. So breakout rooms, they're going to be in groups of 4 to 5. You're going to talk about these 3 things that are in the purple square, and team manager. So team manager today is the person, okay, with team manager whose first name begins with the letter that's closest to the no, I'm going to change it. That's closest to the letter K, as in Madam Vice President Kamala Harris, okay? So if your first name is starts with the letter closest to the letter K, you're the team manager. And so as a manager, that does not mean you do all the work. It does not mean you micromanage everybody. What it means is that you keep your team on track with time. So I would say, like, the first thing should not take your team more than 5 minutes to discuss. Doing the predictions should maybe take another like 6 or 7 minutes tops. And then the majority of your time spent in your breakout room is going to be planning the experiment, drawing the serial dilutions, thinking about what the data is going to look like, coming up with the next and the whys, the next and the whys. You're going to decide who is going to start creating the shared document. Okay, so hopefully somebody with a the ability to share to the rest of the class and not have to write live. So somebody that has something like this, or has a whiteboard that can easily show the class like that, or you type it up, whatever it is. Decide who's going to be the doc composer and sharer. And finally, if the team manager does have good internet connection, I expect you to be the verbal presenter. If you don't, please assign someone to do that. Okay, that's your job as the manager. Okay, and then once we come back as the entire class, I basically just pick random breakout rooms and have you share. It's kind of like DQ. And I add to this working document that I have, and then we just kind of add people's ideas together, collaborate, and then we polish it up so that as a class, we have one very specific, very straightforward problem, predictions, and a planned experiment. And then I'll explain what you do next week. Okay? Any questions on what you need to do in your breakout room? Okay, so welcome back to class, to the entire class. And so what we're gonna go ahead and do is we're just going to open it up to breakout room managers and talk about the research question. So I think for this first one, we don't actually need to see anything. I think that what I will do is I will take notes as the breakout managers share what they thought the research question was. So let's just start, let's go backwards. Let's start with room 6. So the manager, please tell us what you thought what your group came up with as the research question, problem, phenomenon, whatever it is that you came up with.
Hi, can you hear me?
Kinda. It's a little choppy, but okay. Let's see if we can go through it and see what happens.
Okay. Do you want me to share my screen, or should I just read off of my Google Doc and explain what we got?
I think it would be good practice to have everyone kind of take notes on what you're saying, and you don't have to share your screen, but you can obviously use your document. You don't have to have things memorized. So yeah, go ahead and just verbally say it for us.
Okay, so our research question, what we should be looking for is that we are trying to determine the concentration of our stock solution. Sorry, I keep opening up the wrong things. So we're trying to obtain the concentration of our stock solution. So the question is, how do we do this? And the answer should be that, well, from what we know, our rationale is that we know there's a direct relationship between concentration and absorbance. So we can attain the concentration of the stock sample by performing serial dilutions, which will give us multiple different concentrations, give us a wide data set. And then we use the spectrophotometer to measure the absorbance of each of the serial dilutions. And then we should be able to determine the concentration based off of what we know is that there's that direct relationship between absorbance and concentration.
Okay, great. All right, so what I kind of captured on this electronic piece of paper, does that kind of match what you were saying?
Yeah, looks good.
Okay, cool. So let's just leave it there. Let's hear from a few groups and then we'll kind of dissect and perfect. So thank you, Michael. So let's go with room number 4. Hello. You can highlight any differences. You can say some of the same things. You can say how you might change the question. So group number 4, please go ahead, your team manager, verbally share what your group came up with as the research question.
So it was basically the same thing. We were just wondering, why does the color of methane blue become lighter during the dilution? Is the concentration affected by this? So it's basically kind of the same thing as group 6. Okay,
so then if it's the same thing, then just do us a favor, just tell us what the actual question or problem that you came up with was. So if I had to put one thing next to this bullet point, what should I put down?
So our observation or our question was, why does the color of methane blue become lighter during the dilution? Is the concentration also affected by the dilution?
Okay. 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 So the rest of the class should be thinking about these questions and thinking about how we can address them, and maybe can we already answer some of these questions. Okay, so let's go with group number 2. Thank you, Kevin.
Hello.
Hi.
So we just said pretty much the same thing. We're using the spectrophotometer to find the concentration of an unknown stock substance, but we're also using the serial dilutions to create the diluted solutions of stock.
Okay. All right, let's just do one more. Breakout room number 1.
So ours was about after the 5 serial dilutions, what will the rate of change for the concentration and like absorption be for each of the individual dilutions? So like from the first dilution, how will it change over and over and over?
Okay, okay. Does that kind of sum up what you said? So the rate of change between the first dilution, which is actually the stock, versus the last one. So how does the absorbance change as you're diluting each thing one by one? Yeah? Okay. Okay, so I think we've got some stuff to work with here. And this actually is really important because I want to draw your attention to something that I think is very important, and sometimes it is a bit misleading. So remember when we said that we needed to identify the controls, and controls are stuff that you know information about, whether it's actual or whether it's theoretical. Okay. Now, the groups that shared just now are, and please correct me if I'm wrong, but you are all under the impression that you have, let's say you're going to make this serial dilution, however many tubes you need, and you are all under the impression that the goal of this entire activity is to understand what the concentration of each of these is. If that is correct, please put yes in the chat. Okay. Great. So I'm understanding everyone's interpretation of this activity correctly. And the way that you say you're gonna do that is you're going to put each of these into a spectrophotometer and get the OD. Okay, so what if now, let me get my notebook out. And that's why you're telling me things like this stock. Determine the concentration of this stock. Here it is. So what if I told you that this so-called stock had a concentration of 5.86 grams per milliliter? Could you, without sticking any of these in an OD, you still have to do the serial dilution, but without sticking any of those in the OD, could you figure out the concentration of all of these dilutions if you had the original stock concentration? Okay, the answer is yes, right? So the magic word is in theory, right? In theory, if this was 5.86 grams per mil, you would be able to, like Wk2 said, calculate the concentration of this. How would you do that? What's the dilution factor? Just remind me, what's the dilution factor in this activity?
1.5.
Awesome job. 5. Okay. So the DF is 5. So if the first sample was 5.86, you would calculate the concentration of the next sample by doing what? Awesome. Great. By dividing by 5. Okay. So if you divide by 5, you would get the concentration of the second two. If you divide by 5, you get the concentration of the next sample, etc. So what that means, we've already seen them 100 times today. Stop. So what that means is you, in theory, know the concentration of all this stuff. So this is actually not the research question. We know this. We know the answer to all of this. I didn't give you the concentration of today, but I told you that I would give you the concentration. So in theory, we already knew the concentration of stock, which means in theory, we already know the concentration of all of the serial dilutions. Okay, so we actually know all of this. And what did I say the control is? Known information, whether it's actual or whether it's theoretical. So what you all described to me are basically the controls for this entire experiment. So I don't know if you run through it, but there are 3 more samples that are not part of the serial dilution that we don't know the concentration of. And this is why we are doing all of this right here. So with that information, with those serial dilutions, and with the OD that those dilutions give us, that's how we're going to be able to figure out the concentration of these 3 samples down here. Please stop. So now, is there any questions so far? Because I think that that's an important concept that is extremely important to understand. That the serial dilutions have nothing to do with our research question. They are actually our controls. That's what's important here.
can I ask a question really quick, or just kind of a clarification?
Sure.
So are we basically using the serial dilutions to ensure that there is a relationship between the dilution rate and the absorbency? Like we're using this to make sure that we have like a relationship between to make sure that our specta. So we're using that, like I called it an S machine just when I was doing research, but so we're using that to just make sure that we know what we're measuring with before we actually measure the other ones,
Yes.
and that makes it a control.
Yes, in a sense. So you're using the controls for two things. One, yeah, we want to make sure that the spectrophotometer works, okay? And so that every time you do dilute the concentration, so this is going to be more concentrated than that, right, by a factor of 5. In theory, if you stuck this into the machine and then stuck this into the machine should have a value that for OD for this should in theory be 5 times greater than this one. This should be 5 times greater than the next one. This should be 5 times greater than the next one, so on and so forth. And that is based on the previous knowledge that concentration and absorbance of light waves has a direct relationship with one another. Okay, so one, you want to make sure that that is true, but eventually what you want to do is if you have concentration values and absorbance values, can anyone tell me what you do with the concentration and absorbance? So that actually, let's go ahead and move on to the next part of this conversation. So the next part is, now that we've established, so I will recap it here. So we've kind of established that the controls are the serial dilution. Okay. And you're going to use, so I'm really sorry, everyone. This was an unplanned maintenance appointment. I would not schedule this during class. Okay, so the control are the serial dilutions, and you're going to, for each serial dilution, record an absorbance value. And now what you're going to do with that is you're going to eventually, and we're going to get there in this next conversation, you're going to use that to create an equation. And this equation, the entire thing here, this is what will allow you to solve the concentrations of the samples that you don't know the concentrations of. Okay, so predictions and planning. Now that we know what the controls are, what did we say in the beginning of the talk or the class is the controls need to be established first. You need to know exactly what to expect with the controls. So let's see, 1, 2, 4, 6. Okay, so let's do group number 3. This is what I'm going to ask you about the serial dilution. So I'm going to go ahead and stop sharing my screen, and you're going to tell us all about this supposed serial dilution, any information you think is pertinent.
Okay, can you hear me, Dr. B?
Yes, thank you.
Okay, so just to clarify, you're just asking how to perform the serial dilution?
Yep.
Okay,
so
perfect.
If you have like a picture or something that might be helpful for us to see at this point.
Yeah, let me share my doc real quick. Okay, well, I took a photo and embedded it in this document.
Okay,
Okay, cool. So basically you're starting off with a stock solution. So you said in theory it was 5.86 grams per milliliter. And that's going to be your D1, and that's a 1-to-1 ratio. And you have 5 milliliters of that, and you want to, let's see, solve for the volume of the previous solution. And so what you do is just divide that by the DF. So it's just 5 divided by 5, and that's 1. So you're going to be adding 1 milliliter of the previous solution to each of the successive test tubes. And then if you just do 5 mils minus 1 mil, that's 4 mils of diluent. So you should add 4 mils of deionized or water to each of the test tubes. And in the boot camp, I believe you wanted to end up with a 1 to 3,125, whatever. So you need to carry out 5 dilutions. And so basically you just set your micro pipette to, I guess, 1,000 microliters. You'd use the P2000 or P1000 and just add, yeah, and then just add that much solution to each test tube.
Awesome. Yes. Okay. That was explained really, really well. You have the DF on the screen. So the DF is equal to 5, which means every single dilution is a 1 to 5 dilution of the previous. And you explained the math correctly. And I'm going to kind of just copy your drawing on the document that I'm working on, 1, 2, 3. So you have a total of 6 tubes, 1, 2, 3, 4, 5, 6. Okay, so I'm going to have you stop sharing your screen, and then I'm going to reshare mine. We're going to work on this together, or recopy it together, because really it's the same thing that you just said. Okay. So I just want to have it all in one recording. So the dilution, as Joaquin said, the first one is going to be the 1-to-1. And in theory, as I just explained now, the concentration is 5.86 grams per milliliter. And so he wrote these all out correctly. So I'm just going to copy them down on this piece of paper. Okay, and he just, I'm assuming him and his group just took the information from the protocol to come up with this picture here. And so they figured out that you needed to make 5 dilutions, right? This is not technically a dilution, it's the stock, it's not diluted. So you're gonna dilute this. This is the first dilution you make. This is the second dilution, the third dilution, fourth, and fifth. And so he figured out, or they figured out, that you needed to have 6 total samples. And this would be the dilution of each of these samples once you made them. So let's write some other important things. Dilution factor is 5. Total volume when you're making them should be 5 mils. Okay, and I wrote the math over on the right-hand side. So if you are calculating the volume of the previous solution, you need to pipette 1 milliliter. If you are calculating the volume of the diluent needed, or the solvent needed, you need 4 mils. Let me actually write that. Okay, and you just do the same thing every single time. So now what I'm going to ask is, since these are our controls, we need to write down everything we know about them. So in theory, if the first, we're going to do this here in the class, so get out a calculator, whatever calculator you're using, and everyone is going to put this in the chat. So what would be the concentration of the 1 to 5 dilution? Okay, so 1, I'm trusting you. I don't have the key in my head or anything like that. So we're going to say 1.17. Okay. What about the next one? .23. Okay, and so I'm assuming everyone remembers that the way that people are getting this is because they are just dividing that by 5. What if the dilution factor was 10? How would you do it? Just describe to me how you would get the concentration of the next two. The DF was 10. Okay, and if the DF was 4, you divide by 4. If the DF was 100, you divide by 100. Yes, okay. This tells me that everyone has a really good sense of what serial dilutions are. So in here, we're just gonna write etc. And we can keep going. So in theory, we know this. This is why these are our controls. And then you're gonna stick each of these into the spectrophotometer and you're gonna get a piece of data. So let's pretend, I don't wanna confuse anyone. So I'm gonna say, you're gonna get a piece of data here. Okay, so if we go back to what we originally set up here, first things first, create the serial dilutions. So we actually planned that out. We planned out how we were gonna do it. Check, we're done with that, okay? So this is done, done, done. Next, stick it in the spectrophotometer. Okay, so you're planning this, so now you know what the next plan is. The reason why you stick it in the spectrophotometer is to obtain the OD. That's what those little data words mean. Okay, so now, let's see, who else do we need? Group 5. Tell me what you do with this data right here. So team manager, team 5, please tell us, what do we do next? Now that if we were doing this in real life, what would we do with this information?
Hi, I'm the team manager for this group. Can you hear me?
Yeah, and who is this so I can bring you to the front of my screen, at least I can see you.
Yeah, this is Ellery.
Okay, hi.
Hi. I have really bad Wi-Fi right now, so I actually had a verbal presenter assigned, and that was Sierra.
Okay, no problem.
Okay, thank you. Sorry about that.
No worries.
So after we obtain all the data, we would take the OD and the concentration and make a plot where the OD would be your y-axis and your concentration would be your x-axis. And then when you run that through your graphing program, you can get the y equals mx plus b, but it's going to be set to 0, so it's just y equals mx.
Okay, can you tell me what is set to 0 and why we set it to 0?
The y-intercept is set to 0 because you can't have negative concentration. So we will not be going below the x-axis.
Okay, so because negative concentration does not exist, this is the only time in which a scientist should be able to, this is kind of like manipulating your data, but in theory, it is okay in this situation because negative concentration does not actually exist. So if I drew a graph, right, here's your y-axis, here's your x-axis, and Sierra correctly described the x-axis as being your concentration, and the y-axis is being where you would plot the absorbance values. So this is your y-axis, and the b is equal to the y-intercept. So just a reminder for everyone, what that means is you want to know where along this line, if you had some line that was kind of like this or even like this, where does that line, where does your data line cross the y-axis? That is your y-intercept. So anything on this side of the y-axis, that's positive concentration. And then anything on the other side of that axis, that's negative concentration. That doesn't exist. So what we say is that if you had a data line, the only time it would cross the y-axis is when concentration is 0. Because if concentration is 0, how many wavelengths of light are you going to absorb? If there's nothing in there to absorb light, you're not going to absorb anything. So that is why we set the value of b to 0. And this is something that you manually have to do in whatever program you're making a graph in. You can't just add this as another data point. You can't write 0, 0 as another data point. It's something you manually have to do. So thank you for explaining that. So instead of having y equals mx plus b, we can rewrite it to just say y equals mx. Okay, so here's the serial dilution, here's obtaining the OD, and then what do you do with that? You take those values and you graph the data, and then when you graph the data, you come up with the equation of the line. Okay, so let's see, since we're on this group, so Sierra, tell me, what do you do? Remember, everything you do gives you a valuable piece of information to know or to use. So what do you do with this piece of information? Why is this helpful? Why did we need to make this?
You can use this equation to take the OD that you obtain from your unknown samples, plug in that into your equation and solve for X so you can know the concentration of your unknown.
Okay, so yes, but let's fill in all those details so that we have a very good understanding of this. So how can we rewrite this equation so that it has the characteristics or the traits that we're interested in?
You can,
now it's y and x, and so how can we rewrite it?
You could rewrite it as OD equals M concentration, the little brackets.
Awesome. Okay, and so how she got that was she established when you were plotting your data, she said that we were going to put the OD values on the y-axis. And the way that I remember it when you're graphing data is I always remember that the x-axis is the information we know, and the y-axis is information we're gathering. So this is the data that you're obtaining. And that's how you should plot any two pieces of data, is the known stuff's always on the X, and then the Y is the data that you're obtaining. I'm sure there's exceptions to the rule, but I made it through grad school thinking this, and I got published, so I think we're good. So because you've established Y equals OD and X equals concentration, now you can rewrite the equation so that is a little more relatable to what we're doing. And so the spectrophotometer gives us OD values. And so if we go back, remember what our question was. We have these three other samples that we don't actually know the concentration of. Remember, that's our whole question of the day. Okay, so that. Okay, so what we're eventually going to do, I'm not going to tell you, but Sierra says we should rearrange the equation so that we're always solving for concentration, right? That makes sense, because if you look at the problem of the day or the problem of the lab, the question mark is concentration. We're trying to solve, so we want to solve for concentration. And so now we've created this equation that may allow us to do that. But let's rewrite it, because people don't really love solving when things are backwards in the equation. So let's just set this equation so that we rewrite it, and everything that we're solving for is the concentration. So all I basically did was take OD, and then you divide it by the slope. So this is your magical equation. This is what is going to potentially allow us to answer our question. Okay, great job, group Sierra and Ellery and the rest of you, because I forgot what number we're on. So let's go back to breakout room. I don't know, let's do number 4. Wait, was that who you were? 4?
No, we were 5, you're good.
Okay, so let's go back to group 4. So this is where we're at in this whole entire activity. So how do we actually figure this out? What do we do with this information? We have these 3 samples. We really love these samples. We would love to use them in experiments, but we can't do any experiments with them until we know their concentration, right? Because a protocol usually says like, add 50 micrograms of this. And you're like, well, I can't do that because I don't even know the concentration. So what do I do? So what do we do with these 3 samples over here? These are not part of the serial dilution anymore. the whole roll of the serial dilution tubes, they're done, they're out of the scene, we don't even care about them anymore. We used the serial dilution simply to just get this equation. That's the whole point of the serial dilution. So what do we do now, group 4?
So we could just put them into the spectrometer, get their get their optical density, and then we can solve for the concentration using the slope from the previous serial dilutions.
Awesome. Okay, so what we do is we put these in the spectrophotometer. So measure OD of each of these, of each sample. So now you get a piece of data, piece of data, piece of data. And the piece of data that you're getting is your OD. So each of them, like who's talking? This doesn't randomly.
It was me, sorry, Kevin.
Kevin, okay. So then, as Kevin said, stick that value into the equation. So just for an example, let's call this sample A. And let's pretend that the data, just so that everyone knows how to use the equation, let's just say that the OD data was, I don't know, 4.9. Would never happen because the limit of the spectrophotometer is 2, but let's just pretend. And let's say that the slope, when we got this equation in the first place, was just like 0.5, okay? So all you would do to solve for the concentration, this is how I might write it, concentration of sample A. You take the OD, which is 4.9, and you divide it by the slope, which is 0.05, and then you get some answer. What is that, like 100 or something like that? Okay. I don't know what it is. 5 divided by 0.05. Someone help me. What is it? No one? No one has a calculator? Thank you. Okay. And you know from before that in this experiment, we had the concentration units as grams per mil. So we didn't do anything with this. We didn't change it. You could change it if you needed to. If the number was really, really large, you can convert it to kilograms per mil. If the ending value was really, really small, you can convert it to micrograms over mil. And so I think there's some, what are the units of optical density? Yes, it does not have units. Correct, Michael? Yeah, so the way that Beer's law, Beer's equation works out is that optical density, you end up dividing the units and they all cancel out. So this is unitless. Okay, and then a slope, you know, is unitless too. It's just a value. Okay, so that is really great. So this is actually what you're going to be doing. You are going to be collecting this data from the serial dilutions, plotting it onto a graph, creating or generating the equation of that line. Then you are going to use that equation to determine the concentration of your 3 samples that you don't know the concentration of. And then to communicate that, okay, so you're going to be collecting the data, analyzing, and coming up with a conclusion. The conclusions are very straightforward in this study. You are going to conclude what the concentrations are. Okay, so here, where are we? Okay, and this study is the concentration of those 3 samples. Okay. When you're analyzing, you also want to make sure that you analyze the appropriate, that you analyze the trend. So everyone said, here's some of your predictions. That is, concentration goes up, OD goes down. Another way to write that is as concentration goes down, or each dilution, right? Because every time you dilute something, you're lowering the concentration. So each dilution, the OD should go down. Okay, so keep that in mind. And that actually comes back to the second group that presented their research questions is concentration affected by dilutions? And we know that the answer to that is yes, because the definition of a dilution is cutting the concentration, is lowering the concentration. So we actually know this. The answer is yes. This is not something that we needed to investigate in this study. This is already known. Okay. Why does MB get lighter? MB is methylene blue. As you dilute methylene blue, you are minimizing the number of blue molecules. So for each dilution, what you're essentially doing is lowering the number of blue molecules. So if you have less blue molecules, it looks less blue. And if you dilute it again, you are again lowering the number of blue molecules. So fewer blue molecules, less blue to the eye. So those are questions that we actually already know the answer to. Okay. Any questions on this activity or this research problem so far? All right, awesome. So I'm going to share with you the assignment that you are going to do in your groups. Okay. Let me pull it up on Canvas really quickly. Okay. Really good job, everyone, participating and not only verbally, but also in the chat. I really do appreciate that. Okay, so here's the page for week 2. You come here. I hope that you've already come here a couple times this week. And so here's all the stuff that I'm assuming you did before you walked into Zoom Lab today. And hopefully you turned in your worksheet last night. And what you'll find, hopefully those that did the reading, at least for the boot camp part of these things, I hope that you feel that that reading and the worksheet that was due really did kind of put you at a much better place than had you not read the manual at all. And that really is the goal. Okay, during lab, we talked about spectrophotometry. I haven't yet, but I just have a couple things to say about journal articles. You'll find your recording for this session sometime tonight or tomorrow morning. And you can always feel free to look at what was discussed in the Tuesday lab if that's interesting to you. And then end of lab. So you'll be assigned a worksheet. And this worksheet guides you in communicating the findings of this spectrophotometry exercise. Okay, so in order to do this, you need the starting stop concentration. So I have thus given that to you, 5.86, what did I say? 5.86 grams per mil. So now you need that, so continue with that. And you will, in order to obtain the data, this is where you obtain the data from. So if you just click this, we're not gonna watch it here. It's ridiculous. Okay. So it's just a video, 30 minutes long. Paloma, please get in here. Stop. The problem in the plan. Okay, so you can see, I'm not gonna like show things, but yeah, it's me in the lab doing certain things, blah, blah, blahing. Okay, but I actually do the spectrophotometer. I measure the absorbance of all of these samples. I say them verbally out loud. You can see them on the actual screen. So everything is there for you. You record the data. Okay. So you'll watch that. And then here is the prompt for the assignment that's due next week. So next week, Lab Eve, you have 2 things to turn into Canvas. One is all about the spectrophotometer, and they are turned in as separate assignments on Canvas just this time, because this spectrophotometer thing, each person has to do their own. You can collaborate, you can work together, you can help each other make the graphs, you can help each other, you can talk about the conclusions as much as you want, but the writing part needs to be in your own words. And I realize that people's graphs may be the same. That is okay, but the words that they write on the piece of paper need to be in their own words. So if you look at this worksheet, these are the guidelines for how you would write a formal figure. You're not going to draw it. You're not going to plot it by hand. You're going to use a computer program, whether that's Excel, sheets, numbers. These are all the different programs that would allow you to build or to create a formal figure. And so your assignment should essentially just start with a title up at the top. And the title for most scientific publications or reports is generally the take-home message of the study. But in this case, I do not want to see titles where you say sample A is this, sample B is this, sample C is that. That's not a really, I guess, formal title. So come up with a title that either embodies what you did in this activity or is the take-home message. And so this is not your first lab report. You're not writing an intro section. You're not writing the method section. It's just the practice making one formal figure and writing the complementary results to that figure. Okay, and I kind of provide you with some guidelines on what to do. And you can even see by the way that I broke this up into paragraphs on the guidelines, that's kind of how you should break it up into paragraphs when you are writing the actual stuff. So this first paragraph really is about, hey, what's the problem? What's the approach? The next paragraph is what's the data? What's the data mean? That's it. So I'm looking for 2 to 3 paragraphs tops, not pages and pages and pages. More is not more in this class. Succinct, concise, to the point is best in science. Okay, get away from trying to sound super, like use really big words. You don't have to do that in science. No flowery adjectives, just get to the point. Okay, so then you're going to insert a page break. So it's something that you can physically do in a Word doc. You just insert a page break. And on the next page, you should have something that looks like this. It's tiny on this piece of paper. And I did it tiny like this just so that everything would fit on one page for you. But really, when you are making figures, I'm asking you, please don't embed them into, for reports, please don't embed them into your writing. That's why I'm asking you to please insert a page break. This should be the only thing on the second page. Okay, your figure nice and large, and so that you include the important information in a figure, what I am going to draw you to is the expectations for lab reports in general. Okay, so I give you a couple pieces of information. I even told you like, hey, this is how I made this little bunk figure. I just it's all made up, right? I just made up these things and I put it here so you can see what one would look like without giving you a real graph with real data, because I want you to be able to work through this on your own with your own data. So if you go over to the modules and you go up to the place where I said like, hey, here are the general requirements for formatting. I think it's here. Let me see. Okay. Yeah. Okay. General requirements for formatting. The last kind of section is all lab reports. No, this is not a full lab report, but it does have information on how to write or make a formal figure. So if you go there, you go to the part where it starts talking about what goes in a figure. Okay. Okay. Doo-doo-doo-doo. Okay. Here we go. All right, so here's some information on what makes a good figure, what makes a good title for a figure, what makes a good key for a figure. So I'll leave that there for you to read and use so that you can do this assignment. Okay. Okay, and then where you submit the finished product, the assignment link will be in next week's page. And I think I already actually made that for you. Let's see if I did. Week 3 lab. Yep. Okay. So before week 3 lab, yeah, I think I almost finished week 3's page. So here's your here's your assignment. Once you're all done with it, you just click here and that's where you submit it. But you'll also see that next week, week 3 eve, you have something called your JC notes. So what does that actually mean? JC notes. So if we go back to this week's lab, and what did I say we were also going to talk about in this week? In the second part of the lab, I will introduce the purpose and the significance of journal articles. And you can kind of see that that is all highlighted by purple. I like color coding. Okay, so at the end of lab, which is now, you're gonna be assigned to read a scientific journal article. So I'm not gonna go into a lot of details about why scientific journal articles are important, but I'll just basically say, this is how scientists communicate their hard work to the scientific community. And then other scientists use that as their previous findings to do the next big thing in the field. And so these journal articles, they're called journal articles, they're called research articles, they're called scientific publications, they're called primary literature. Those terms are all synonymous. All of them, if they are of good quality, will have been peer reviewed, which means if I'm ready to submit my information with words and beautiful pictures and this nice story, I submit it to, it's kind of like a newspaper or like a journal, and you want those people to publish your work. The journal editor doesn't just say, cool, publish next month. They say, well, first, let me make sure what you're writing here is legit. So you're writing about what? Brain research? Cool. I'm going to go contact 3 scientists that are pros in brain research too. I'm going to give them your publication, and they're going to give you a very true, accurate, kind of sometimes harsh assessment of your work. And that's where the whole scientists don't beat around the bush. They don't need to sugarcoat things. They're just going to tell you how it is. And so they each write a report back to the author, and they say, like, this was crap. Or the second reviewer might say, I love this, but this one experiment didn't really convince me. So you need to either redo it and get better data, or do a different approach to get more convincing data. And so only when all 3 of those reviewers come back and give you a thumbs up, only then can your research article get published. So to put it in perspective, to gather enough data, at least as a molecular biologist using mice as a model, took about 4 years. 4 years to get enough data to write it up in a manuscript and send it to a journal. From there, it took 10 months to get published, because the reviewers take X number of weeks to read it, send you back things that you have to do. Typically, it's more experiments. Then you have to rewrite it. You have to make new figures. You send it back, and they do it again. So it is a pretty laborious process. So when you do get a peer-reviewed research article published, a scientist will know they have good faith in that. So that is all I'm going to say about journal articles for now. And they are the bread and butter of like what makes you a good scientist. People always want to know, what journals did you publish in? How many publications do you have? It kind of says like, all right, are you just sitting in the lab making a million mistakes for 10 years? Or are you hustling and producing some pretty interesting information? Okay, so you're to have an experience reading a research article. And this may be cumbersome, scary, overwhelming for a lot of you, or maybe you're excited for it, or maybe you're a pro and you don't even need this. So for those of you, well, for all of you, I've created a how to take notes, a guideline, and this can be used essentially for any journal article, but I wrote this one with the journal article that you're going to read in mind. Okay, so this is here for you. So when you do read the article and take notes, you should use this as a guideline so that you know you're taking detailed enough notes. Right? So kind of break it down, and this goes hand in hand with some of the articles that you read coming into lab, like how to read a research article. So take this, and this is your choice. So you were in a breakout room with however many people, 4 or 5 people total. From there, you can work with a team of 2 or a team of 3. So if you were in a team of 5 for today, you can split up into a team of 2 and a team of 3. Or you can choose to do this by yourself. So this is your option. So no matter what, you have to turn in notes. So if you work as a team of 2 or 3, only one person in that team needs to submit their notes to Canvas. So it's almost like if you're a team, you're one person. Only one person on the team needs to submit to Canvas. Just write everyone's names at the top of the document. And also when you submit it to Canvas, write it in the comments box. If you choose to do this by yourself, you are more than welcome to, and you'll turn in whatever you have as well. Okay. And you don't need to tell me who's on each person's teams beforehand. You just figure it out on your own. So you have that at your disposal to use. And I even give you an example. So once you kind of figure out like the overall ideas and the kind of the background information and kind of like the overall methods, then you should dissect the figures one by one. So I kind of break them down for you. So you should break down these into figure 1, and then for figures 2 and 3, you can see that break it down into figures 2A through C. What is the question that they're asking that is being shown in these 3 panels? So a panel is panel A, panel B, panel C. You should not have 3 questions for each, like one for each panel. Consider panel A through C the entire figure. What is one question for that entire figure? So I break them down for you so that it's a little more digestible. And then I wrote an example of how to do this for table 1. So that's there for you so you can kind of get an idea of what I'm talking about when I say like, hey, for each figure, what's the research question? What's the approach? And what's the answer? So I do this for table 1. Okay. And then here is where you find the actual paper. Okay. And so this is a slightly different version of a research article. Research articles are typically like very, very, very long. This is a letter. So anytime something is a letter, this is a letter for a journal article called Nature. So a letter is kind of like an abbreviated, summarized version. And so, yeah, if you look at this right away, you're going to say like, that is a lot of words. It is. So there's also tables and pictures. Okay. So I wouldn't start trying to write notes when you start reading this for the first time. Here is my piece of advice. What I would do is I would look at the first sentence in each paragraph and write that down as if you were writing an outline. So if you can put this sentence in your own words, and it may require you to do some online research. Maybe you don't know what dopaminergic means, go look it up. Maybe you don't know what the striatum means, go look it up. If you can understand the first sentence in each paragraph, then you can go back and start writing notes. So first sentence in each paragraph creates for you an outline. And then you can start filling in with more information. This stuff in bold right here, this is your abstract, okay? After you've read the first sentence in every paragraph for the paper, go back and read the abstract. The abstract is the summary. It will have a couple little pieces of information about the background, a couple pieces of information about what we already know. Hidden in there somewhere will be the objective, the goal of this paper. And then finally, the end of the abstract should have the take-home message. That is an excellent abstract. It just a little bit from each section. What do we know? What background information do we need to know? What's been done? What do we do? What did we find? That's it. Okay. And do realize it will seem like a foreign language to you the first time. That is totally normal, totally okay. And the more you work with this, the more you will understand. Okay. All right. Oh yeah. And then the last thing that I did was not only did I write a demo of how to take notes on a figure or a table, I actually created for you a presentation, a little bit about journal articles. If you were presenting them in a PowerPoint version, how would you do it? So I actually do this with the introduction of the paper and table 1. So it's another YouTube video. I would try to at least read the paper once before you watch this video. That would be my recommendation. Okay. Any questions on the journal article or taking notes on it?